In our final lecture in the organic chemistry series, we're going to talk about separations and purifications. Starting first with solubility-based methods. These methods use the solubility of certain compounds in the respective separations. The first type is an extraction. This is the transfer of a dissolved compound from a starting solvent. It uses a separatory funnel, and once the product is extracted, the extraction solvent is then evaporated. A wash, on the other hand, is a reverse extraction. This is used to remove impurities by removing what is not desired. If you don't remember what a separatory funnel looks like, it looks something like this. So what you're going to see is a funnel with some sort of dropper apparatus, and you're going to see two layers, generally an organic and a solvent, an organic and an aqueous layer. Depending on which one is denser, the denser layer is going to sit on the bottom, and your respective desired compound is going to be in either of the layers. What you do is you shake this up, pour out either the desired layer or the undesired layer, and then extract the remainder. What you do for a wash is you take your desired layer and wash it through with the non-layer. So if my desired layer is organic, I'll wash it through with water to get rid of any polar impurities. Filtration is the isolation of a solid from a liquid. So with an extraction, you're dealing with a liquid-liquid compound, but for filtration, you're looking at something that is a solid that has not completely solvated into the liquid solution. What you do is you trap it into a filter, and the desired product is the filtrate. The product is then washed and purified subsequently. There's two types we'll go into. Gravity filtration uses the product's own weight to pull through the filter, whereas a vacuum filtration pulls the solvent through via a vacuum. If you've ever seen a coffee filter, it relies on the same principles. You keep the grounds in, and you let the liquid go through. Recrystallization. This is a method for further purifying crystals into a solution. The product is dissolved into a hot solvent and is allowed to recrystallize as it is cooled. In order to increase the purity of the product, what you'll generally do is pick a solvent in which the solid product that you desire is only soluble at high temperatures. Because of this, as the solvent cools, only the product will recrystallize out. At the bottom is an example of this recrystallization. You'll see some very beautiful crystals because of this. If you're curious, this is how crystals form in general. Now that we've talked about solubility-based methods, let's go into distillation. A basic overview of distillation is in order. For starters, distillation takes advantage of differences in boiling points to separate liquids. How far apart these boiling points are is going to dictate which type of distillation method you use. The collected evaporated solvent condenses into a distillate. Below is an example of a simple distillation. The impure liquid is boiled, and the evaporated desired product is pulled through a column and allowed to condense through. What comes out at the end is our distilled liquid. There are different types of distillations, like I mentioned. The first is a simple distillation. This is used with a distillation apparatus and a cooled column to collect the distillate. The image prior is an example of a simple distillation. If the boiling points of the two solutions are similar, then you can use a simple distillation. However, when things get more complicated, we have to use a different method. A vacuum distillation uses a vacuum to lower the pressure, thus decreasing the temperature required to boil the liquid. This is used when the two liquids have very high boiling points. It makes it so that we don't have to achieve as high of a temperature. Fractional distillations use a fractional column that forces the liquid to evaporate and condense at various levels. This reflux ensures that only desired product is collected. This is used when the two liquids are similar in boiling points. If you don't remember what a fractional column looks like, it looks something like this. So if on the inside of the column, you will see ridges, very similarly like so. As the product and possible impurities evaporate up, it's going to cool via this area right here, this outside area. So there's going to be some sort of cooling mechanism going through here. It's going to cool, and liquids are going to collect in these areas. Well, once it's going to continue to get heated up and continue to travel up, collecting and collecting until eventually only the desired product is pulled out. When two liquids are very similar in boiling points, you have to use something like this in order to ensure that you have what you want. The last bit of instrumentation we're going to go into is chromatography. Below is an example of a gas chromatograph. This is based on Leonardo da Vinci's designs. Obviously, because of the time in which he was alive, he didn't really have the technology to create something like this, but he did theorize its existence, which is very remarkable considering the time he lived in. The process is simple. You run a mobile phase through a stationary phase. The characteristics of the substances in the sample will dictate how they elute through the stationary phase, and the migration distance of each substance is calculated and partitioned. As you can probably guess, different compounds have different partitioning coefficients. The first type of chromatography we're going to go into is the thin layer and paper chromatography. This is where known compounds and samples are spotted onto silica plates. You can also use paper for this, which is something you've probably done in your general chemistry lab. The mobile phase is then run through the stationary phase, and the migration distance is calculated to determine the compound in the sample. This is based on the retardation factor, which the equation for is listed below. To the bottom right is an example of a chromatography. As the solvent front rises, the mixture is going to separate accordingly. If the solvent is more polar or less polar, it's going to determine the flow with the solvent front. Take a guess as to what type of solvent was used here. The top one is less polar, and the bottom one is more polar. You guessed it. The solvent is probably some sort of organic, because the less polar compound stayed with the solvent longer, whereas the more polar compound came out earlier on, suggesting that it had less of an affinity for the solvent. By doing this, you can determine what sort of compounds are in a mixture. Column chromatography uses a column filled with some sort of media, usually silica beads or some sort of aluminum compound. The varying degrees in which the sample's compounds interact with the column media will dictate how they flow through the column, and partitions of the samples are collected at different times. Depending on what you desire, you could also use this as a separation method. There are three types of stationary phases that are commonly used. These are referring to the beads that you see in the columns themselves. The first type is an ion exchange. This is where beads are coated with some sort of charged substance. This binds oppositely charged compounds. As you would guess, if the charge is the same as the compounds in the stationary phase, they're going to come out faster than the ones that actually bind to the compound. Because of this, sometimes if you make the affinity strong enough, you can actually collect a desired compound from just washing it out of the beads. Size exclusion utilizes beads that contain pores of various sizes. This allows compounds smaller than the pores to pass, whereas larger compounds cannot pass, making them move faster through the column. This seems very counterintuitive, but the reason why larger compounds move faster is because instead of going through the bead pores, they go around them, meaning that it goes through at a faster rate. This one trips up a lot of students, so make sure you have that distinction straight. Lastly, affinity chromatography utilizes compounds of interest by creating a medium with high affinity for the compound. What you do is you create the medium, usually some sort of protein receptor, that will attach to the compound of interest. This is how you can collect it through. Washing it out of the column media is how you get through to this. So, in a way, you can use chromatography as a means of separation. The next two methods we're going to go into are a little bit more complex forms of chromatography, and they're often used with a computer. The first is gas chromatography, or GC. This is also known as vapor pressure chromatography. The compound is vaporized and injected into a column. The vaporized compound then travels through the column at varying rates. As you would guess, the column has a very specific composition, and how quickly or how slowly the vapor gets through this column is going to determine largely based on the compound. The speed at which these come out of the column are then measured via a computer, and the waste is just discarded. The last instrumentation we'll go into is high-performance liquid chromatography. This used to be called high-pressure liquid chromatography. However, with modern technology, we don't have to have as high of a pressure to run this anymore. It's okay if you slip up and call it high pressure though. I know I still do that. But a more correct definition for it is performance liquid chromatography. In this method, the liquid eluent travels through a column of defined composition. This allows for very specific solvent gradients and procedures, thus leading to a higher degree of precision. Below is an example of this, where one type of solvent is used, but note that you can use multiple different kinds of solvents and have them go in at partitioned specific compositions and rates. This can further enhance your data and make it more precise when you're trying to figure out compositions. Note that this is similar to gas chromatography, but it uses a liquid medium instead of a gas. Fantastic. So we have covered the entire organic chemistry series. These are subjects that you will likely see on the MCAT. So make sure you study up, and we'll see you in the next lecture series.